of where RNA polymerization needs to occur. How does it know which promoter to engage? How does it know to bind to that promoter? That is the job of the sigma factor, which can be swapped out, and different sigma factors engage different types of promoters. Once that sigma factor hits that promoter, it says, okay, this is a good place for us to just get started. Let's associate in a more permanent way at this particular site. At that point, then, the DNA double helix gets unwound, and we create this so-called open complex, and we begin establishing this transcriptional bubble, this open part of the DNA in which the bases can be exposed and used as a template for RNA synthesis. Once that open complex is established, we actually don't need the sigma factor anymore, and it can wander away, and the core polymerase of RNA polymerase can begin to do its job in terms of elongation and generate this RNA product. Just to show you what that transcriptional bubble looks like again, we just need to pop open a little bit of the DNA double helical DNA double helix to provide for the template strand to be used to associate the incoming nucleotide triphosphates, right? And the reason that we have a template strand and a coding strand, you think that they would kind of be synonymous. So if you want to read the sequence that's going to be made, you can read off the coding strand, but what's actually being used to provide the template and what we form hydrogen bonds to with the incoming monomer units is the template strand. And then there's two exit channels. One exit channel is for the reformed double helical DNA, and the other exit channel is for the product RNA. All right, so how do we terminate? And as I mentioned before, there's two ways that RNA polymerase knows it's done its job and come to the end of the region that is supposed to encode for mRNA. One of these is the topology of the RNA product itself, the fact that in some cases it generates this hairpin-like structure, this stem loop structure. It's rich in GC base pairs. And the formation of the product with this kind of topology is sufficient for the RNA polymerase to know that it needs to disassociate and complete its synthesis. The other is the function of this protein complex rho helicase, which chases along after the polymerase. And if it catches up, it pushes that polymerase off the DNA. Here is another picture of that stem loop type structure. And so we see that, yes, it's rich in these GC base pairs, and it's got a bunch of U's on the 3' side of that stem loop structure. And then a little bit more detail about this rho protein, which is a motor protein. It's a helicase, and it runs along. It sits on the RNA product, and it sort of plays this game with the polymerase as far as whether or not it can catch up and engage with the polymerase and disassociate it. This is an ATP-driven motor protein, right? And this thing works because of the fact that RNA polymerase does not move at a constant rate down the DNA template. In some cases, it can move very smoothly and speedily, and in other cases, as it's generating different types of RNA product or as it's hitting different sequences within the DNA, it actually begins to slow down, can even stall a bit, and that functions as an internal signal, allowing that rho protein to catch up, and as it does that, it can push that polymerase off. So the termination depends on the strength of the RNA pause signal that would be here. That if it's weak, then we need that rho signal, and if it's a strong indication from the topology of the product that RNA polymerase needs to stop, we actually don't need that rho effect. Okay, so that concludes really our evaluation of the role of RNA polymerization in eukaryotic systems. The fact that we have these two different types of way to terminate. We'll talk towards the end of the semester about mechanisms for gene regulation. How do we turn genes on and off? We'll talk about that in particular with regard to eukaryotic systems where this becomes quite complex. We'll talk about how proteins can bind around the promoter region. I'll talk a little bit about that today and more about it towards the end of the week. Because under some circumstances, we want some mRNA, we want some protein expressed as the cell responds to different conditions. So we need regulatory elements that are around that promoter. Sometimes they're quite close by, and actually sometimes the binding of protein factors, even fairly far from the promoter, can alter the ability of the polymerase to sit down in front of the start site for mRNA synthesis, and we call these factors, these proteins, we call them transcription factors. Some of them are promoter proteins, and there's other proteins that actually block the polymerase from engaging, and we call those repressors, and we'll talk quite a bit more about those again later in the semester. So let's begin to think about this process as part of the larger picture of both transcription and translation, and think about the differences in a prokaryotic system and a eukaryotic system. So with regard to prokaryotes, it's actually quite straightforward. The polymerase, reads off the DNA template, makes the mRNA, and then even as that mRNA is still being generated, the ribosome can begin to use the mRNA as a template to generate a protein product. So these two different facets can be completely coupled, coordinated, and can take place simultaneously. That's not the case in a eukaryotic system, where the synthesis of the RNA takes place in a physically different part of the cell than ribosomal-mediated protein synthesis. So in this case, transcription takes place in the nucleus, and there's a lot more processing of the RNA that needs to happen. We generate a mature messenger RNA product, which we'll talk about today. That mature mRNA then gets exported through the nuclear pore out into the cytoplasm, where it can then be read out by the ribosome. So we need to understand this process in order to figure out what is really going on in a eukaryotic system prior to thinking about the translation of the mRNA message into protein. Okay? So prokaryotes, transcription and translation, coordinated, can take place at the same part of the cell, and in eukaryotes, these are segregated. Another aspect that makes transcription more complicated in eukaryotic system is just that the template, the DNA itself, is a lot more structurally complicated than in the case of a prokaryotic genome. We talked before about how the DNA in eukaryotic systems needs to be highly compacted in order to fit within the cell. The role of histones and forming nucleosomes that allow that condensation. And this occludes the DNA from being accessed by the polymerase. So part of the process needs to be to open up this condensed chromatin, to decondense it so that it can be accessible, to remove the DNA from its complex with the nucleosomes, and then to engage the polymerase along with a whole bunch of other protein transcription factors to allow the generation of the RNA product. So all of this adds to the complexity of the transcriptional process in eukaryotic systems. Questions? Okay. So let's think about polymerases. Let's think about regulatory sequences that would be around the start site for RNA synthesis. Typically, we think about a region upstream of where we want to begin our mRNA synthesis that contains a promoter region. And typically, we talk about a TATA box that contains the sequence TATA. This was thought to be the canonical region that provided a promoter sequence for most of the genes in a eukaryotic system. We now know that there are a bunch of different types of promoters. TATAs are used very often, but not all the time, not even probably a majority of the time. As far as the enzymes that are generating the RNA polymerase, there's 3 families of RNA polymerases, Pol-1, Pol-2, and Pol-3. And they're called upon to generate different types of RNA products. In general, polymerase 1 binds to promoters upstream of where we want to generate ribosomal RNA species. Pol-2 is usually involved in synthesis of mRNAs, and we'll be talking a lot about polymerase 2 today, looking at its structure and thinking about how it engages its promoter. And polymerase 3 generates transfer RNAs and also the 5S ribosomal RNAs. So again, we're going to be focusing in on polymerase 2. It's much more complicated than the polymerases that we talked about in the prokaryotic system. It contains 12 subunits, but its largest subunit is similar to the beta subunit of the polymerase of E. coli that we looked at before. And we'll look at these big multi-protein complexes that are created to form the active polymerase. The 3 different types of polymerases, again, they give rise to different cellular transcripts. They're also been characterized biochemically by the fact that they have variable responses to a toxin that is an inhibitor of some of the polymerases. So this is the toxin amanidin, comes from the death cap mushroom, and the mechanism of its toxicity is specifically to shut down the activity of polymerases. Here's the complicated structure. It's kind of cool. But yes, so some of the polymerases are inhibited by that toxin and others are not. Here's a little bit more information on those 3 different polymerases, polymerase 1, 2, and 3. Again, these respond to different types of promoters. And you can see here is, again, the TATA box type promoter that's usually used to engage RNA polymerase 2. And again, these are used to generate different types of RNA products. So let's take a look at this big complex of proteins that begins to engage the DNA and think about the same steps of initiation prior to the generation of the RNA product. So here is our coding region. That contains an initiator, sometimes abbreviated as INR. And then upstream of that, there's that promoter that's so typically used by polymerase II, the so-called Tata box. And so this constitutes what's called the core promoter region. Lots of times there's additional regulatory sites that are upstream. They can be fairly close by, or hundreds, even maybe thousands of base pairs upstream. So the ones that are closer by are called the enhancer regions, and additional regions can also be present that bring in these transcription factors and activators that allow the polymerase to be associated with the promoter region. As I said, the Tata box was the one that was initially described and the one that's best known. There are particular transcription factors that bind onto that particular promoter. This TBP actually stands for Tata box binding protein. Other transcription factors are transcription factor 2D that binds there. But you'll see that there's other types of sequences upstream of the start site that can engage other transcription factors and are also used to encode, to direct the initiation of the polymerase for other types of gene products. We now think that that Tata box is responsible for providing a promoter for about a quarter of genes in humans. Oftentimes there's multiple upstream promoter sites. A very simple one might be here for beta globin. Again, typical polymerase II promoter with that Tata box, and then one additional site, the CAAT sequence. Here we see that CAAT sequence right there, so-called CAAT box. Other viral proteins might have multiple promoter regions upsite that don't involve a Tata box. And you can see that there's different types of arrangements of these that can extend a few hundred base pairs upstream of that start site. Activator regions can be really far removed from that core promoter region. Here's one example of an enhancer element that's binding to an activator protein really far away, and it's actually bending the DNA and allowing this so-called mediator complex that we'll take a closer look at later on in the semester. And then that helps to recruit the polymerases to come into that core promoter region. And we'll talk, when we discuss aspects of regulation of gene expression, we'll talk about how different activators can be used to recognize different sequences. All right, so here's the process that corresponds to the initiation of RNA polymerase in eukaryotic systems. You'll see that the polymerase itself, the enzymatic core, actually binds pretty late in this process. So first we get this like Tata box binding protein and perhaps other transcription factors that associate just upstream of the initiator region. Then we bring in the polymerase. Helicase activity is required to create the open complex, and this creates that transcriptional bubble that we were looking at before in the prokaryotic system. And at that point, we can phosphorylate a region of the polymerase, hyperphosphorylate that region, and that provides a signal for the polymerase to actually begin moving down the DNA template and begin cranking out the RNA product as shown here in green. The polymerase, at the same time that it's carrying out its main task of generating the RNA product, it's actually performing a surveillance role as well. It's actually looking at the DNA and trying to figure out if there's any DNA damage, any sites where nucleotides have been cross-linked to each other, for example. At that point, the polymerase will go, wait a second, I don't know what to do right now, and it will stall at that site. And that will be a message for the cell to bring in repair mechanisms that can come in and actually fix a lot of the damage, chemical damage that's been done to the DNA template. So the polymerase assembly plays two roles in generating an RNA product and in providing surveillance for DNA damage. This shows the critical protein-DNA interaction that's right up here at the start of this process, this Tata box binding protein, TBP, sitting down on that region of DNA. And this just shows that that is a dimeric complex, and the interactions between the protein and this particular sequence of DNA are mediated by two phenylalanine residues that engage with the DNA on either side of the double helix and read that particular sequence. And we'll talk more later on in the semester about the precise mechanisms that protein-DNA interactions can lead to sequence specificity. Exactly what is the protein looking at that's allowing it to bind to some sequences versus the other. Once the Tata box binding protein hits that particular sequence of DNA, again, it begins this whole process of recruiting all these different transcription factors in advance of the DNA polymerase itself binding, and this just shows transcription factor 2A as being one of the first steps in that process. Another just reminder about these regulatory elements. Some of them are proximal elements, within, let's say, 100 or a couple of hundred base pairs upstream from these promoters. And then others are enhancers that can be located quite distant from the promoter site. And we saw this example of a long-range interaction that alters the DNA geometry or topology itself. Okay, so once we've got everything in place, and the DNA polymerase, the RNA polymerase itself is part of that active complex, we can begin to generate the mRNA product. And it's a very similar process to what we were looking at before in the case of the prokaryotic systems. And this shows you the transcriptional bubble that's really very similar. DNA, double helical DNA comes in, it's disassociated, creating that transcriptional bubble, and then the DNA template strand reassociates with the coding strand, and the double helical DNA comes out of the exit tunnel. You can see right here that there's a small element within that transcriptional bubble where we've created a duplex between the incoming RNA nucleotides forming the RNA product, and it's just a little region right here where we have that RNA-DNA duplex right in the middle of that polymerase. The action of RNA polymerase can be inhibited. So I mentioned to you before that the polymerase is looking for damage within the DNA, cross-linked nucleotides, for example. So the polymerase is actually fairly sensitive to alterations in the geometry of the substrate. And inhibition of the polymerase can also be caused by binding of small organic molecules into the DNA double helix. We took a look before at these so-called intercalators, these aromatic small molecules that can sit within a DNA base pair, and these, some of these are shown right here. This acridine, this is actinomycin D, and these sit in the DNA, they bend the DNA, and they block the movement of the polymerase. And so these types of molecules have been explored and used as anti-cancer drugs. So why do you think that these types of molecules that would inhibit polymerases, would be particularly good as chemotherapeutic agents? Yeah.
Slow down cell division.
Okay, so these are going to be selectively toxic to cells that are undergoing rapid division. So those sorts of cells will be particularly dependent on the generation of both mRNA and also ribosomal RNA. So if we can shut that down, we'll have selective toxicity to rapidly dividing cells. So here's the process for elongation. This is the C-terminal domain of the polymerase. As you can see, it's depicted here as being phosphorylated. That's the signal for it to be undergoing the process of elongation. And it begins to generate an RNA product. And as it's generating that RNA product, already some of the processing of that RNA begins. One of the very first things is that the 5' end of it gets a cap. And so we'll take a look at the nature of that cap in just a bit. But the 5' end of the RNA product is chemically altered. And then as the RNA begins being generated, we're going to begin the process of removing the non-coding portions of the RNA. And so that is the excision of these introns, and we'll talk more about that in just a bit. Then, as that mRNA product is produced and completed, one of the last steps in that is the generation of an altered sequence at the 3' end, where a whole bunch of adenines are tacked onto that 3' end. That provides a signal for the polymerase itself to terminate, and the RNA polymerase disassociates from the DNA and then can be used again to generate a new mRNA product. This shows some of the processes that are used in generating the mature mRNA. So here we initially generate our immature transcript, our precursor mRNA, and then we can either bring in special machinery to remove the non-coding intron regions, or we can count on those introns to actually splice themselves out using a couple of different mechanisms that I'll talk about in just a bit. Once we have that mature mRNA that's got all the introns removed from it, it can be exported through the nuclear pore out into the cytoplasm and used as a substrate by the ribosomes. So here are the overall descriptions of the sorts of modifications that are made to RNA to process a primary transcript into the mature product. A 5' cap is put on, a 3' polyadenylate region is put on. The termini can be removed. Sometimes there's non-coding sequences at the end in advance of the polyadenylate. Introns are spliced out, and then oftentimes bases within that RNA and sometimes sugars are chemically modified. We'll see this is particularly the case for tRNAs. The mRNA can then be used as a template by the ribosome until such point as it's no longer needed, and then the mRNA can be degraded.
Here's what the 5' cap of the eukaryotic mRNA looks like. Okay, so that's the region that gets put on right here. This is a 7-methyl guanosine, right? So here's our 7-methyl guanosine position right here. It's added through a really unusual linkage onto the 5' end of the RNA. It's actually a triphosphate linkage that extends from one 5' hydroxyl of the RNA polymer product to the 5' hydroxyl of the 7-methyl guanosine position. And then also sometimes this 2' hydroxyl on the ribose rings at the 5' end is also methylated. Let's spend some time figuring out how to get rid of those introns. So I showed you some basic aspects of this before. The fact that very typically in eukaryotic RNAs, the initial product that's made has these huge regions in it that don't code for protein and that need to be removed. And very often, there's actually more non-coding regions, these introns, than coding regions, which we call exons. You can actually make electron photomicrographs that really indicate this in a kind of a beautiful way. If you take a mature mRNA and then hybridize it to the original template DNA, you're only going to get hybrids formed where you have the exons present. And so you can see that in this case, this electron photomicrograph has got all these loops associated with it that correspond to these intervening regions. So even in this really typical gene of albumin, you can see that there are 7 introns within this thing that need to be removed. In some cases, the introns are actually able to carry out this process on their own. And so there's a couple of examples. Group 1 and group 2 introns, both are capable of directing their own splicing. And we'll see that in the case of group 1 introns, they actually bind a guanine nucleotide, and that provides for the initial nucleophilic attack that begins the process of excising this intron. And then we'll see in group 2 introns, it's actually an intramolecular nucleophilic attack that provides this first step. So here's the process for a group 1 intron. In this case, the initial nucleophilic attack comes from the 3' hydroxyl from guanosine or guanosine monophosphate, diphosphate, or triphosphate. And that's the hydroxyl group that makes the first attack at the 3' end of the exon in intron junction. So does that seem as though it would be a typical nucleophile, asking the 3' hydroxyl to play the role of a nucleophile in this system? Is that what we see, for example, in the case of polymerization? Is that nucleophilic attack carried out by the 3' hydroxyl? Anybody? So I asked you to consider, in both the case of DNA polymerase and the case of RNA polymerase, what the chemical steps are that gives rise to the product. Okay, so the attack by the 3' hydroxyl is the typical process that occurs, okay? So this seems completely normal. And it's being carried out by an exogenous nucleotide that's bound in at this intron. Once we have that initial nucleophilic attack established, we've generated a new 3' hydroxyl right at that site. Now we have that 3' hydroxyl at that exon. That can be used as a nucleophile to carry out another attack on the other end of the intron. And it's at that point where we religate the backbone of the RNA, generating the spliced RNA and excising the intron, okay? So that 2 nucleophilic attacks, 2 esterification reactions have to occur in order to remove that intron at that site. And this just shows that 3' hydroxyl undergoing that initial attack. Here's the incoming exogenous guanosine providing that attack, and we call this a transesterification because we start with an ester and the product is an ester, but we've just swapped one ester for another. Okay, so it's this exogenous guanosine that is coming in and conducting this reaction to form this new ester linkage. A similar process happens for group 2 introns, but in this case, it's all intramolecular, right? So we need to break that phosphodiester bond at that upstream site, at that exon-intron junction. But now, in this case, it's actually going to be a 2' hydroxyl of an adenine residue right in the interior of the intron that's going to carry out the formation of that first ester linkage. So at that point, we generate an intermediate where we have that adenosine that's actually got 3 phosphodiester bonds. It's got the typical 3', 5' diester bonds, and now it's got an additional phosphodiester bond at the 2' position as well. Once that initial reaction has occurred, again, this is the same as group 1 in the fact that there's 2 transesterification events. We've generated a new 3' hydroxyl that can be used as a nucleophile to loop out that lariat type structure. And at that point, we've generated our properly spliced RNA, and that intron is now gone. One difference is that in this case, we've generated a linear product from the intron, and in this case, we've generated this loop type structure of this lariat that's been generated. In addition to self-splicing introns, sometimes we need to bring in an additional piece of machinery to cut out introns. These can be formed both by proteins and by RNA molecules. We call these spliceosomes. And again, these associate with the intron, carry out the same type of esterification reactions on one end, and subsequently at the second end of the intron region, and these also generate these lariat type structures. So I'm so enamored of these lariat structures that I'm showing you this again. I think this is really cool that we've got these particular adenine branch points, and I'll just point out again that we've got our phosphodiester linkage, typically at the 3' and at the 5', but we also have another one at the 2' position, and that's the point where it comes all the way back around to form this loop type structure connected back at the 5' position. Okay, so we've put on our 5' cap. We've removed all of our introns, and now we need to process the 3' end and add our polyadenylate sequence. There is a typical 5' sequence, AAUAA. An endonuclease comes in and clips after that position, and that forms the proper recognition site for an enzyme called polyadenylate polymerase to come in. It accepts ATPs as substrates and adds them one at a time to the growing 3' end, and it's actually a pretty long polymer chain, all of adenines that are added to that 3' end, anywhere from 80 up to 250 adenines are added to make the mature mRNA product. Now, I want to point out that on the one hand, this polyadenylate polymerase is doing something that should seem perfectly normal to you guys. It's taking nucleotide triphosphates and it's adding them on to the growing 3' end of a nucleic acid polymer product. No big deal. The thing that makes this polymerase unusual is that every other polymerase that we've been talking about requires a template, but this polyadenylate polymerase does not, right? So it's just taking the mRNA and adding to it. It knows exactly what to use, only incoming ATP monomers, and so it actually doesn't need a template to decide from one position to the next what nucleotide triphosphate to use. So here's the overview of what happens in mRNA processing. We have our DNA, right? It's got both coding and non-coding regions. Can be pretty large. Let's say for the ovalbumin, 7,700 base pairs are provided for in the gene itself. The RNA polymerase comes down onto that template and generates the primary transcript. It's got both coding and non-coding intron regions in it. The 5' cap is installed. Then splicing occurs through, typically through those group 1 and group 2 intron splicing events. All the introns are cut out of that primary transcript. The additional RNA at the 3' end is removed by the nuclease. Once that region is removed, the polyadenylate 3' tail can be added at the end, and then we generate our mature mRNA product. The overall process then begins with something that might be 7,700 bases long and process it to the mature mRNA, which can be substantially smaller than that in the case of ovalbumin, 1,872 nucleotides. So this is very straightforward. There's one gene, or ovalbumin, that encodes one mature mRNA product to make one particular protein, ovalbumin. We've commented before that it's remarkable that there aren't too many genes in the human genome, but there's actually quite a number of protein products. So the question then becomes, how do we get a bunch of different protein products when we don't have that many genes? And a big part of the answer is that we can process these primary transcripts very differently to give rise to different mature mRNAs, which are going to encode for different proteins. So here's one example of that. This is an mRNA that encodes for a couple of different peptide hormones. We generate this primary transcript, but in this case we can have two particular positions at which we add a polyadenine sequence. We can either cleave right here after the fourth exon, or after the 6th exon, and add our polyadenylate after either of those particular regions. So in this case, we'll get one mRNA that has the first 4 exons, and in the case of generating these mRNAs in brain tissue, we're going to create a mRNA that has information from 6 exons. We can then translate each one of those, process the initial protein product with a protease to give rise to two completely different peptide hormones, either calcitonin or calcitonin gene-related peptide. And these both play different roles within the body. So from one gene and one primary transcript, we get two different mature mRNA products, and two different proteins are generated from that one gene. So that's just one example of how a primary transcript can be processed differently. In this case, it's two different sites can be cleaved and polyadenylated. These sorts of variations in mRNA splicing can be carried out in a number of different ways. So here at the top, you see a representation of, let's say, a really typical processing of an mRNA, but we can bring in alternative promoters and begin the process of RNA synthesis at an alternate start site, in which case we're going to generate a truncated mRNA that's going to be missing the first part of the canonical product. Talked before about different places for polyadenylation. We can skip over a particular exon, so we generate a smaller mRNA product that way. We can leave an intron in and not excise it. There's different ways to splice at the 5' or 3' end of an intron, and each of those creates a slightly different mRNA product. So all of these can be brought to bear to take one gene that can be read out in one way and use it to generate slightly different protein products. In addition to the modifications of RNA that we're talking about with regard to this typical processing, individual bases and sugars within RNA products can also be modified. And in some cases, we're talking about methylation onto the nucleobase itself. And in other cases, there can be modifications onto the sugar, such as an alternate position on where there's an attachment site onto the ribose. We typically see these modified bases in tRNAs, and when we talk about how tRNAs are used as an intermediary in reading out an mRNA product on the ribosome, we'll talk a little bit more about tRNA structure and the role of these modified bases. Okay, any questions about RNA processing? All right. So I'm now going to flesh out a little bit more about the use of nucleic acids as templates for polymer synthesis. And I'm going to spend just a little bit of time talking about ways that information flows in some kind of unusual ways. So in the case of reverse transcription, we're actually pushing information backwards. We typically think about DNA encoding RNA, but reverse transcription has information flow backwards, in which RNA is used as the template for DNA synthesis. And then we'll also see one example from viral systems in which RNA encodes its own synthesis that can be used for replication of an RNA genome. So these kind of exceptions to the central dogma typically come up in the case of viral systems. And in the case of reverse transcription, we see these for retroviruses. So retrovirus is a virus that has its genetic information stored as an RNA genome. And in this case, in order for the virus to successfully infect its host, an RNA genome is not going to do it a whole lot of good because it won't be properly recognized by the host cell. what the virus wants to do is to integrate its genome within the host cell chromosome. And so it needs to make this important first modification, which is to read out its genome in the form of DNA. So after being taken up into the cell, the RNA genome is subjected to reverse transcription and is converted into a DNA product. Once that happens, that viral DNA can then be taken up into the nucleus and inserted into the host chromosomal system. Reverse transcriptases are important targets in antiviral pharmacology. And reverse transcriptases are also really important in molecular biology techniques and in DNA cloning, because very often what we want to do is make a cDNA, a copy DNA, in which we subject messenger RNA to reverse transcriptase, generate a DNA product, and use that as a component in cloning. Why would we go through the effort of taking a messenger RNA, converting it into DNA, and then using that for cloning? Why wouldn't we just go to the DNA itself? Yeah.
The fact that it's in the RNA form means that it's being expressed. You might be interested in genes that are being expressed at the current time, as in they're being expressed in the protein.
Okay, great. So if what we're interested in doing is like some kind of analysis of gene expression levels, then what we really care about is not whether or not a particular sequence is present in the genome in the form of DNA, but actually how much of it is present as an mRNA at any particular point in the cell cycle or in response to different disease states. And in order to quantitate that, it might be convenient to use the reverse transcriptase. But in terms of cloning, let's say we wanted to do expression of a particular protein in a bacterial host. It becomes really important to generate cDNA and to use this reverse transcriptase to make a DNA copy from the mRNA. So why would that be the case? Yeah. Right. Right. So let's say we have that chicken ovalbumin gene, and we want to generate a huge amount of that protein by expression in a bacterial host. So we can turn to the chicken DNA, the genomic DNA material, and use that, but that's going to cause all kinds of problems because it's got all of those introns in them. And so if we just use that DNA, inserted it into a plasmid, and then use that information to encode for a protein, that bacterial cell is not going to be able to process that properly. And it's going to use the initial RNA product and to take it to the ribosome, and it will generate an incorrect polypeptide that includes all of that material within the introns as well. What we really want to do is create a DNA version of the mature messenger RNA that's already been processed to remove all of those introns. So the best way to do that is to obtain the messenger RNA, and that's actually fairly easy because that polyadenylate region can be used as a hook to isolate out mature mRNA products. Then subject that mRNA to a reverse transcriptase, make a DNA copy of that very conveniently has all of those introns removed, and then use that copy DNA inserted into a plasmid. Now we use that as a template for expression in the bacterial host, and we get the proper ovalbumin protein product generated. So in that sense, these reverse transcriptases are super helpful, and we use them all the time. Great, so that's one example of an unusual flow of information. And then, as far as replicating genomes, this is usually what we think of, that genomes are composed of DNA, and in order to get replication, we need DNA-encoded DNA synthesis. But there are some genomes that are composed of RNA. So these retroviruses have an RNA genome. And in some cases, these viruses take advantage of RNA-dependent synthesis of RNA. So we see this in bacteriophages that infect E. coli, and the enzyme that we call upon to carry this out is an RNA-dependent RNA polymerase. We also call it an RNA replicases. And this is a really interesting example of how pernicious or clever viruses can be, that although it needs this enzyme to replicate its own genome, it actually grabs almost all of the machinery for doing that from the host. Only one of the 4 protein subunits that's used in making this polymerase is actually encoded for by the virus itself. The virus grabs proteins that are already present in the host cell and uses that for the majority of creating these polymerases. That polymerase then has a strong preference for replicating the viral RNA, right? So if the virus just made an RNA-dependent RNA polymerase and it was indiscriminate, that machinery would end up making a whole bunch of copies of all the other RNAs that were present in the cell. Instead, it has a strong preference for replicating the viral RNA template. And there's one other example that I know of a ribosomal RNA from Tetrahymena that's been shown to polymerize RNA onto an RNA template. Why do we care so much about this process? I mean, it's kind of interesting as an exception that proves the rule to the typical flow of information in biochemical systems. But there's another important reason, and that relates to the RNA world. So why do you think this would be so important when we think about RNA as being like the primordial biopolymer? Why do you think this might be so crucial? Yeah.
If there were no way for RNA to self-replicate, then if it were primordial, there would be one generation and it would die.
Right. So when we take a look at modern biochemical systems, we're looking at different biopolymers to play completely distinct roles. We look at DNA to provide a permanent source of genetic information. We look at proteins to be the functional molecules within the cell and carry out enzyme catalysis, for example. And we look at RNA as providing an intermediary in that process. When we think about the origin of life and we want to consider what the very simplest life forms would be like, we are hunting around for one biopolymer that can do it all, right? That can be a source of genetic information, that can carry out catalysis, and that can replicate itself. That's a lot to ask for. Proteins for sure can't do that because they can't be replicated, right? There's no way to use a protein sequence as a template. DNA seems completely inadequate in carrying out that role because DNA is not catalytic. It lacks the functional attributes. But this is one additional example of how RNA, in some sense at least, can carry out all those roles. We know RNA can be a repository of genetic information because we look at RNA viruses. We know that RNA can be catalytic. I just showed you these self-splicing introns, and we'll see other phenomenal examples of catalytic RNAs in the form of the ribosome itself. And now we see that RNA can even catalyze its own replication. At least in a rudimentary way, it can polymerize ribonucleotide triphosphates. So it does seem to be a good candidate for the origin of life. All right. So this kind of fleshes out these exceptions here in green to the typical flow of information, and indicates that RNA can also be used as a template for nucleic acid synthesis. The final step in this process then we'll begin considering next week, which is how RNA can be used as a template for protein synthesis, the process of translation. We'll take a really careful look at what happens on the ribosome, but we can begin to think about these issues right now. And it's a very interesting historical issue. Your textbook talks about the RNA tie club, when the early researchers understood that DNA was the repository of information and that somehow it needed to encode for protein, but the question was, what was that process like? And the questions began pretty quickly, like, okay, we see the sequence of nucleotides within DNA. How do we associate a particular sequence of amino acids in a polypeptide chain? Do the amino acids actually physically interact and recognize DNA as the template? And it'll see a stretch of DNA and say, okay, I'm a tryptophan monomer. I'm going to sit down on this DNA and allow myself to be polymerized at that position. What was the physical mechanism that we could read out a nucleic acid sequence and get it into a polypeptide sequence? So they began asking these really fundamental questions like, okay, how many nucleotides are there that encode information in DNA? 4. How many amino acids are there? 20. Okay, thank you very much. So how do we encode that information? Can we make a one-to-one correspondence between a nucleotide sequence and a protein sequence? Does that seem possible? No. If we made a one-to-one correspondence, we could only encode 4 different amino acids. How about a 2-to-1? How about 2 nucleotides encode 1 amino acid? Is that going to do the job?
No.
Yes, no. I see a big fat no. How many amino acids could be encoded for by a 2 nucleotide sequence? 16. 4 squared, 16. So at a minimum, we need a trinucleotide sequence. How many units should we be able to encode with 3 nucleotides? How many different ways might there be to read out 3 nucleotides? 4 times 16 times 4 is 64.
Yeah, but 61.
So now we're seeing this discrepancy between 64 units of information and 20 amino acids. So now we're thinking, gee, that there's some kind of mismatch there. What's the nature of that mismatch? And once again, how does a particular amino acid read what presumably has to be 3 nucleotides on the DNA? It doesn't seem likely that the amino acids are going to be engaging the DNA directly. So that was the impetus for a lot of initially just hypothesis. Francis Crick hypothesized that there must be some type of adapter molecule that must physically engage with the nucleic acid, read out a triplet, at least a triplet, and then somehow associate that adapter molecule with a particular amino acid. And we will see how tRNA nicely plays the role of that adapter molecule. As we begin to think about translation, we will begin to think about which triplet codons are going to be assigned to each of the 20 amino acids. We'll think about how those experiments were actually conducted and how the information was obtained. It's really easy to take a look at this genetic code and say, oh, okay, that's the way it must be, but somebody needed to figure this thing out. And then we're going to need to evaluate how we ensure fidelity in this process. We talked about fidelity with regard to DNA replication, the fact that the cell has all these different mechanisms to make sure that it doesn't make mistakes, and that if it does make a mistake, it can correct them. So what are the molecular mechanisms that the cell uses to make sure it does not make a mistake in these systems? And we'll see that in addition to this genetic code, there's also what's been referred to as a completely separate second genetic code, and that's the code that specifies how to attach the proper amino acid onto the right adapter molecule. And so we'll be taking a careful look at the family of enzymes that we call upon to carry out those reactions and how those enzymes make sure that they do their job properly. Because it's important that we don't make mistakes. We want to make sure that we're not lost in translation. And I think that's a very fine point for which I should leave you for the Thanksgiving break. I'm going to try to post this recipe for a whiskey apple crumble pie. One of my favorite Thanksgiving pies, and it's what I'm going to be making over the weekend. For those of you who want to work over the weekend, we will have that practice midterm posted and available. Otherwise, enjoy a great meal. And I'll see you next week.